everyone. Thank you very much for joining us in this second talk from the Nova webinars. This is an event organized by the members of the NovaSpec project, a transdisciplinary research project focused on innovations in communication in humans and other animals. This is the second of four very interesting talks that will take place over the course of 2 weeks with speakers from different countries on different themes in communication. Please follow our Instagram profile to get more information so you do not miss any of the talks. Today, we are very pleased to welcome Dr. Robin Zigitella. She is an associate professor in the Department of Biological Sciences at the University of Denver, where she also holds the role of associate chair. Her research centers on evolutionary and behavioral ecology, with a focus on social interactions between the sexes. She investigates how behavior drives rapid evolutionary change, how selection pressures and non-adaptive processors shape communication, and how environmental change influences sexual selection. Her lab also explores the role of parental effects in adapting to new mating environments and how these dynamics contribute to reproductive isolation. Her main research themes include rapid evolution, sexual selection, and speciation, parental effects, behavior plasticity, and responses to global change. Dr. Chingitella, thank you very much for your presence here today. After your talk, we will have 15 minutes to take questions from the audience. Everyone can ask questions through the YouTube chat. Questions could be in Portuguese. Pessoal, vocês podem fazer perguntas em português. Our team will select and change them to me, and we can translate. A gente translate, ó. A gente traduz para a professora Chingitella. With no more details, I will give the word to Dr. Chingitella now. Dr. Chingitella will be here in the background in case of any technical problems.
Okay. Can you see slides okay?
Yeah, perfectly.
Great. Well, thank you so much for the introduction and for the invitation to come speak with you all today. Today, I want to tell you a story about when, how, and why entirely new cricket songs are evolving and spreading in Hawaii. Some of the most spectacular and diverse traits that we find in nature are the signals that animals use to communicate with one another. And we know that closely related species often differ really dramatically in these signaling traits, despite being really similar in other traits. So on this slide, I'm showing you some examples of signals that are used in mate attraction that differ between closely related species in a variety of different taxa. From tree hoppers on the far left, whose vibrations are super diverse, to cichlids in the middle that come in lots of different color patterns, and to cricket songs on the right that differ in their pulse rates. By definition, animal communication takes place when a sender, like this male peacock spider, sends a signal through the environment to a receiver. That signal might arouse the receiver, intimidate a rival, attract a mate, or repel an intruder or predator, for instance. And the receiver, on the other hand, can learn things from those signals, like the sender's species, their identity, location, reproductive status, or even what they taste like. I want you to notice also that this particular receiver is wearing these special glasses, which is meant to indicate that features of the receiver, like their sensory systems and inherent biases or preferences that they might have, should shape which signals evolve and are successful. But where do these signals come from? How do they evolve initially? And what shapes those signals once they arise? Well, we know a bit about the last of those questions, what shapes signals once they arise. You could imagine, for instance, that signals that make animals too easy to detect by predators, or those that aren't easily detected within the environment, might be selected against. Sexual signals like those that I alluded to on the last slide are the very traits that are used in communication between mates. They seem to evolve relatively quickly, they differ between closely related species, and they can act as important barriers to mating. Sexual selection has even been called an engine of speciation. Yet our understanding of the earliest stages of sexual signal divergence is really hampered by our inability to directly watch that process. Instead, we're always looking back on this process, making comparisons between closely related species that already differ in their weird and wild displays. And I would argue that how new sexual signals arise, persist, and spread is really difficult to envision if signalers and receivers co evolve. And that's because if receiver sensory systems and their preferences are aligned with signals that already exist, when a novel trait initially invades and is rare, virtually all receivers should discriminate against that. But what if we could watch the origins of novel signals? What features of signalers, receivers, evolutionary history, and the environment would allow these novel signals to really get established? Well, sexual signals are often complex stimuli that integrate multiple different sensory components. And each of those is produced by underlying morphology, physiology, or behavior of the signaler. So today I want to conceptualize that as the instrument here. And that instrument is going to generate a signal that's broadcast out into the environment. And once they're out there, sexual signals are subject to many and varied selective pressures. So when we're thinking about how novel signals become established, we should be thinking about those selective pressures. First, of course, signals should be amenable to the preferences of their intended receivers. Those are often females who use these signals to make mating decisions. But we should also consider the broader social and ecological context in which they're delivered. So things like competing signalers and transmission properties of the environment that might vary from place to place. The conspicuousness of sexual signals also makes them subject to the unwanted attention of eavesdropping natural enemies, things like predators and parasitoids that literally listen in on these signals and use them instead to locate hosts and prey. And of course, we also need to think about the even broader non-adaptive evolutionary context in which signals have been shaped. So things like historical population relationships and gene flow and genetic drift that really establish the variation that's present within populations and thus how they can respond to future selection. The really elaborate acoustic signaling systems of crickets, which we'll talk about today, are a really great example of the role that animal communication can play in diversification. Male crickets produce songs that function both in species identification and in mate choice. They use a calling song to attract females from afar. And then once they're in close proximity, they switch to a lower intensity courtship song in those one-on-one interactions. And it turns out that the songs of closely related species differ in spectral characteristics and in temporal patterning, which is what you're seeing here. And those differences are the major pre-mating isolating barrier in several groups of crickets. The instrument that these animals use to produce their signals is their forewings. And they rub the file, which is this modified wing vein with a number of evenly spaced out little teeth on it, against the scraper of the opposite wing. So you get a burst of sound each time the wings close. There are also a number of important resonating structures that I've colored here on the wing for you, things like the harp and the mirror that project that sound out into the environment. So let me set the stage for you a bit for the story that I'm going to tell you today by taking you to the Hawaiian Islands. Hawaii is a paradise for a lot of reasons, but for biologists, I think it's a magical location where we can actually watch evolution unfold before our eyes. Lots of really well-known examples of rapid evolution are set in Hawaii, including, for example, the famous rapid adaptive radiation of Hawaiian Drosophila from a single common introduced ancestor to hundreds now of species across the island chain. And on very isolated islands like those in the Hawaiian archipelago, almost everything you encounter was ultimately introduced at some point. When it floated there on waves or swam or flew, was blown in by the wind, or dropped on the islands by traveling people or birds. And of course, we know that introduced and particularly invasive species can cause major problems when they interact with and outcompete natives. But more broadly, when organisms are introduced, they're exposed to novel selective pressures like new environmental characteristics, natural enemies, food, and competitors that they don't encounter elsewhere in their range. And those novel selective pressures can drive the kind of rapid evolution that my lab is really interested in. So this set of characteristics, I think, has led biologists to think of the Hawaiian Islands as the setting for a sort of unique and ongoing series of evolutionary and ecological experiments. Today, then, I want to tell you about the evolution of brand new cricket songs in Hawaii and what we're learning from them about how new animal signals arise and persist. For about the last 20 years, I've been studying male song in this cricket, the Pacific field cricket, Teleogryllus oceanicus. The cricket is actually native to Australia, but it's also island hopped through the Pacific as they accompanied Polynesian explorers on their outrigger canoes. Eventually, they arrived in Hawaii and were introduced there sometime between 700 and 1600 years ago. In this system, then, the cricket's wing is the instrument, and it produces a signal, a loud, long-distance calling song that sounds something like this. That song, then, is subject to these different selective pressures that we talked about, including intended receivers, in this case, female crickets, who use these songs to locate males and to determine which males they want to mate with. In Hawaii, but only in Hawaii, the cricket also encounters a deadly eavesdropping parasitoid fly called Ormia ocratia. That fly is attracted to the songs that male crickets produce to locate females. They'll actually locate a singing male and sort of spray larvae on and around that singing cricket. Some of those will burrow into the body cavity of the cricket, where they'll spend the next 7 to 10 days feeding on tissues inside of the animal before they dig their way out of the cricket, pupating and killing their hosts in the process. We also, of course, work on the environmental considerations that I talked about before, things like social factors and transmission properties of the environment. So we'll talk about those just a little bit today. The overarching question that I want to address today then is why and how has this ancestral signal generated by the wing instrument that's pictured here really diversified over just the last 20 years, generating what we now think of as sort of a rainbow of different types of male song in Hawaii. So this story then is going to start when the crickets were introduced to Hawaii sometime between 700 and 1600 years ago. Our parasitic fly arrived in Hawaii around 1990, so about 35 years ago. And shortly after that, Marlene Zook started studying interactions between the cricket and fly and had really continuous field studies running throughout the 1990s. During that time, all of the crickets had that typical wing type that I showed you before, and they produced what we now call the ancestral or typical song, which you just heard. That song looks something like this picture here. It's loud, it's relatively pure tone, and it has a really characteristic temporal pattern with this trill followed by sets of paired pulses. So I'm going to play that song for you again here so you can really get that typical song stuck in your head. Now, if you know anything about this study system, what you probably know is that in about 2003, we discovered silent male crickets on the island of Kauai. Those males went silent when an X-linked mutation wiped out most of the structures on their wings that are used to produce song, leaving the wings flat rather than ridged. In fact, that silencing mutation actually evolved 3 times independently on 3 neighboring islands. Not only that, but it spread through one population to more than 90% of males in fewer than 20 generations, because being silent protected these males from that parasitic fly. And then things were quiet for quite some time out there in the field. That is until about 2017, when I traveled to an island I hadn't been to before called Molokai. There, I found that the crickets were singing a brand new song, and we called this song purring because I initially thought the strange sound I heard must be coming from some nearby cat. The wings of purring males are really similar to those of silent males. They're missing a number of important structures, and they have really, really reduced resonators. But purring males have an important little piece on the wing called the scraper, and silent males don't. And that means that when purring males rub their wings together, sound is produced. That song, though, bears little resemblance to the typical songs. The temporal patterning is the same. It's a trill followed by sets of paired pulses, but purring songs are quieter, they're higher in frequency, and they're much more broadband than a typical song. Here's what they sound like. Having found that purring song, then, about 6 years ago, my lab started a longitudinal study in which we monitor crickets and flies every 2 cricket generations in 6 different Hawaiian locations. So on each trip, we collect adult animals using methods that are not biased with respect to what their wings look like or what their songs sound like. And then we do a really standardized set of studies looking at sound characteristics, the wing instrument. We also measure the preferences both of female crickets and of female parasitoid flies. And more recently, we've even been measuring their ability to hear these different novel signals. And this approach has been really, really fruitful. For instance, just 2 years after discovering purring crickets in 2019, we started to hear things in our data that we couldn't explain, songs that didn't neatly fit into these existing categories of purring, silent, and typical. And that's because it was yet another new type of male. This one we called rattling. Here's what a rattling song sounds like. Like purring, those rattling songs are quieter, they're higher in frequency, and they're more broadband than the ancestral typical song. But unlike purring, if you just look at the wings of typical and rattling males, they're nearly identical. That is until you flip that wing over and examine the little teeth on that file on the underside of the wing under a microscope. When we did that, we found that the file of these rattling crickets has characteristic gaps in it where teeth are missing but should exist. And those gaps and the resulting spacing of teeth dramatically change the signal properties. Meanwhile, that same year, Nathan Bailey's lab in Scotland published a paper reporting on 2 other novel cricket morphs in Hawaii, each of which produced yet another different song using changed wing structures. One of those we call small wing. It has these miniaturized wings, and the other is called curly wing, that has wings that curl up on the edges, leading to some sort of strange interactions between that file and scraper mechanism. More recently, things continue to evolve out there. For instance, we just characterized yet another new type of male called broken heart. And we'll keep watching and hopefully be able to fill in some more of this story along the green arrow here in the coming years. So this leads us to the idea that what once was a sort of dichotomous, you're either silent or you're singing system, is now one with a rainbow of different types of males that are all producing different songs across Hawaii. For instance, this is a map we made of a snapshot in time showing the dramatically different makeup of our 6 replicate populations across Hawaii. And their different makeups here are indicated by the differently colored slices of pie in each location. The composition of those populations is also highly dynamic over time. So here I'm showing you fluctuations over just 3 years in a single population. This is actually from one lawn that contains 4 different types of male. And between 2018 and 2021, for instance, this curly wing morph in yellow arose or arrived there and then spread to comprise more than 40% of the population, apparently managing to outcompete some of the other novel morphs and even that ancestral type. And the diversification of wing structures and songs is ongoing. Even more recently, we've uncovered loads of undescribed wing variation in samples that we pulled out of our -80 from the past 5 or 6 years of collections. So we're finding things like wings that have forked files or extra veins in the harp, or files with loops in them, and even files on parts of the wing where they don't belong. And while I know these pictures probably don't look like much to folks who don't spend their time analyzing cricket wings, this variation is much, much greater than the variation in wing structures that you typically see even across different cricket species. In fact, last year, Jay Gallagher, a former PhD student of mine, scored about 300 wings that he pulled from our freezer for the presence or absence of 13 traits that are found on the wing. And then he used a hierarchical clustering mechanism to group males based on how similar their wings were. And when he did that, he found 38 different wing phenotypes. Many of those that are signified here in yellow were undescribed types. So are all of those in yellow novel morphs, and does each of them generate a unique different song? Probably not, but I think this undescribed variation is really striking nonetheless. Okay, so we are all biologists here. You must be wondering at this point, what makes each of these new male morphs that produce funky new songs successful enough to invade populations of crickets that have been singing that typical song for millennia? To get at that, we used our longitudinal data set to ask how each described morph balances the conflicting natural and sexual selection pressures that they encounter. So in other words, we asked our cricket and fly receivers how they responded to these novel signals. And we did that using field-based phonotaxis experiments, where we just broadcast different types of song so that we could capture natural mate location and host location behaviors of crickets and of female parasitoid flies. And I'm going to summarize what we found and reported across several different papers in this one relatively simple figure. So on the top, what you're going to see above the axis, the x-axis in light blue, is the percentage of female crickets that are attracted to each male signal type. And on the bottom in dark blue is going to be the percentage of parasitoid flies that are attracted to each of those signals. On the far right, for instance, is our typical song, which is very attractive to females, but also very attractive to parasitoid flies. And on the far left is that silent male type, which completely escapes parasitism, but also has a really difficult time attracting mates. The novel signals are in the center of this figure. And largely, we find that they all seem to be at least moderately attractive to females, despite some of them being really quiet and broadband. And each of them protects their bearers to a large degree from detection by that parasitoid. Some of these even appear to be private modes of communication, at least for now. Clearly, then, how receivers respond to novel signals is going to play a big role in how these songs evolve moving forward. And broadly, to be really effective at eliciting a response, a signal needs to propagate successfully through the environment and then be detected and distinguished from other sensory stimuli by the receiver. Anything that falls outside of a receiver's range of sensitivity is essentially invisible, inaudible, and odorless. It's undetectable. Often, greater sensory stimulation translates into stronger preferences. But how that plays out when the receiver is making a decision really depends on perceptual processing that isn't always straightforward. And there have been a number of recent calls to really better integrate our understanding of sensory and cognitive mechanisms of receivers into our understanding of the evolution of animal communication. So for the rest of the talk today, then, I want to dig in a little bit to share some really recent data. One story about our crickets and one story about the unintended receiver, that parasitoid fly. Like most animals, these two have a really broad sensory capacity that allows them to detect and respond to cues of predators and prey and conspecifics, for instance. And the sensory system really defines the limits of behavioral responses that can act on signals. So, our cricket on the far left here is capable of hearing sounds that range from about 2 to 100 kilohertz. The figure that I'm showing you here is a behavioral audiogram that shows you how loud a sound needs to be at each of those frequencies along the x-axis in order for a cricket to hear and to respond to it. So the lower that black line is, the more sensitive the cricket's ear is to sound at that frequency. So it makes sense then that crickets seem to be most responsive at 5 kilohertz, because that's the dominant frequency of that typical male song, which is the gray spike that's shown here on this figure. On the right-hand side of the slide then is a fly neural audiogram. It's in light pink, and that neural audiogram is indicating the amplitudes at which flies from Florida, where these guys are native, can detect each of those frequencies that are on the x-axis. So same deal as the cricket audiogram. The auditory tuning of these flies from Florida also peaks at about 5 kilohertz, which matches the song of their local host cricket, which is the red spike here. Now, I showed you this figure just a few slides ago, illustrating that most of these new songs attract a really good chunk of the female crickets in Hawaii from at least a meter away. Those data are all from Hawaiian crickets specifically. So females from those locations are relatively accepting of these novel male types. But I want to ask why they're so accepting. It turns out that this was actually a pattern I had seen before. Way back in graduate school, when we discovered silent crickets, I did this big courtship study where I simulated secondary contact between female crickets from across their range and silent males from Hawaii. And what I found was that females from island populations, the black bars here, and particularly from Hawaii on the far right, were much more willing to accept silent males as mates than were females from Australia, where the crickets are native. And this was a pattern that we kept seeing over and over and over in this system. In the study that this particular figure comes from, we tested how females responded to different versions of male purring songs. And we found that across that acoustic space that these new songs inhabit, Hawaiian females were really permissive or accepting. So this preference function or fitness surface is really flat. There was no obvious song, no obvious purring song that was preferred or discriminated against. Females were equally accepting of those. And that pattern that we keep seeing over and over led us to wonder whether relatively relaxed sexual selection might be a hallmark that was facilitating the evolution of novel signals in this system. And if so, what mechanisms lead to that permissiveness? Were they evolutionary mechanisms or were they plasticity? We could imagine that selection might contribute to relaxed preferences in Hawaii in a couple of different ways. And the first I want to bring up is actually related to the colonization of islands. In the 1980s, Ken Kaneshiro hypothesized that in really small, recently founded island populations, search costs are actually high because mates are really rare. So females that have relaxed mating requirements would be favored by selection. And the idea was that a really, really choosy female might never find an acceptable mate amongst the founders, and her genes would leave the population. So this particular pattern is of interest to us here because our older studies suggested that females from island populations were the ones that were more accepting, at least of silent males. We could also imagine there being more recent selection for relaxed preferences in Hawaii because that parasitic fly preys on the loud males, and some of these populations have been primarily or fully comprised of silent males for more than 20 years now. So again, search costs are really high. It could also be, though, that plasticity actually favors those relaxed preferences. There are lots of things that differ across populations of these crickets, like the acoustic environment and mate availability, and both of those could induce plastic changes in mating behavior. So, to test for processes that might lead to relaxed mating requirements in Hawaii, Gabrielle Welsh, a PhD student in the lab, did a really cool big study across their whole geographic range. She started with field-based mate location trials in 6 populations, 2 from Australia, 2 from islands in French Polynesia where the fly is not found, and 2 from Hawaii where the fly is found. And this really broad geographic approach allowed Gabrielle to address different hypotheses about how mating preferences have become relaxed in Hawaii. Things like Kanashiro's effect on islands versus subsequent selection in Hawaii only. And then she did something really clever. She brought eggs from all 6 of these populations back into the lab, and she reared them in common garden and then performed the very same mate location assays on 2nd or 3rd generation lab animals. And that comparison between what happened in the field and what happened in the lab allowed us to ask whether these patterns of relaxation in Hawaii are evolved or are they largely plastic. So what did we find? Well, on the x-axis here is the region where the animals from Australia, French Polynesia, or Hawaii, and the y-axis is the proportion of females that were phonotactic to the different songs that we played for them. And those songs are going to be indicated by colors. We used a silent negative control. I'll show that in black. And then we played 2 novel songs, purring and rattling. Those are in peach and red. And then finally, we played the typical ancestral song, which is in green. And broadly, what we find is that Hawaiian females were indeed the most accepting, but also preferences were very plastic. There were some really interesting differences in the shape of preferences between regions and between lab and field that gave us some hints about why Hawaiian crickets might be so accepting. So I'm going to layer this figure on for you. What I'm showing you so far is that in the field, females from all 3 regions responded relatively the same to the silent negative control and to purring songs, and they didn't really like purring very much. But when we looked at that novel rattling song, Hawaiian females were more phonotactic than French Polynesian and Australian females. And for typical song in green here, all 3 regions differed. Hawaiian females were again the most attracted to it, followed by French Polynesia and then Australia. How about in the lab? Recall that our test for plasticity was whether there were differences in behavior between lab-based and field-based tests. And really interestingly to us, when we tested those females from the very same populations, but did that in common garden in the lab, we found no differences in phonotactic behavior among regions. So let me break that down for you a little. To better visualize these detailed differences in behavior between lab and field, the plasticity, we made a heat map where darker shades indicate bigger differences between the field and the lab settings. So the plus and minus signs on this figure indicate the direction of that difference. And you'll notice that the darker boxes are really concentrated up in the left there. So plasticity was most pronounced for females who are from Australia and French Polynesia, and most pronounced with respect to those rattling and typical songs. Both of these populations, so Australia and French Polynesia, were more phonotactic to those rattling, and typical songs in the lab than they were in the field. And in contrast, Hawaiian females in these light gray boxes down at the bottom of the figure exhibited little plasticity, and they were consistently very responsive to these songs. So they maintained high responsiveness regardless of whether it was lab or field. So, what do those patterns suggest? Well, first, they suggest that there's quite a lot of plasticity in phonotaxis for French Polynesian and Australian females. But perhaps that there's been an evolutionary loss of plasticity in Hawaiian populations that contributes to crickets there being consistently highly responsive or having relaxed preferences when we observe them both in the lab and in the field. And we actually suspect that that loss of plasticity in Hawaiian populations might reflect the fact that they recently colonized a novel environment where preferred mates were rare. And in that case, then, the loss of plasticity allows them to retain those relaxed preferences that facilitate success in this environment by making them phonotactic to pretty much any signal that you play for them. Okay, in the last 5 minutes or so, then, I want to talk a bit about the parasitoid flies. So I'm going to switch gears and tell you a bit about the unintended receiver here. Really influential fields like receiver psychology and sensory drive and sensory bias tend to assume that the sensory mechanisms of receivers are rather conserved, either because of the nervous system or of environmental constraints. But if receiver sensory systems are instead evolutionary labile, this could actually change the dynamics of coevolution when novel signals evolve. The novel songs that have arisen recently in Hawaii have really different acoustic properties than those of all known cricket hosts for the fly. And those differences are in directions that should make the new male morphs more protected from that parasitoid. That is, unless the parasitoid can actually evolve in response. So in this study, we used complementary behavioral and neural experiments to ask how the fly fares first when it has to invade a new habitat in Hawaii, where they had to adopt a completely new host species, and then when that host signal started evolving. And this is work by my former master's student, Erin Weichel, that was done in really, really close collaboration with Norman Lee, who's pictured here from St. Olaf College. So first, what we did was to test for evolved differences in the auditory sensitivity of the flies. So we recorded extracellular neural activity from the neck connective tissue, which contains auditory neurons that send auditory input to the brain. And we tested responses to pure tones that varied in frequency from about 2 to about 40 kilohertz. So that's on the x-axis here. And then we played those tones at sound intensities from about 20 to 90 dB SPL, which is on the y-axis. And we actually did this with flies from 2 populations. One of those, Florida, in the sort of orangey color here, is where the animals are native. And then the second population is from Hawaii, where the fly was introduced about 35 years ago and where these novel cricket songs are evolving. So again, the Hawaiian animals will be in kind of gray-blue here, and the Florida animals will be in that light orange. And the super cool result here is essentially that the shape of the fly's neural frequency tuning curve differs between these populations. In particular, we found that Hawaiian flies are more sensitive to frequencies around 5 kilohertz and around 10 kilohertz. And those are really cool frequencies because they happen to correspond to the dominant frequency of the typical T. oceanicus song and the dominant frequency of the purring T. oceanicus song. Next, we tested for evolved differences in the behavioral response thresholds of Hawaiian and Floridian flies. And we did that using phonotaxis on this really cool spherical treadmill system. So we tethered the flies on top of a floating mouse ball and we played songs to them. And when they responded positively, they would run on that ball toward the sound that they liked. And in these experiments, we found similarly that Hawaiian flies were more behaviorally responsible or responsive overall at a number of different frequencies, but especially to those frequencies that were aligned with the typical and the rattling T. oceanicus songs. But does all of that actually translate into differences in host location ability? So in one final experiment, we did natural song playback. So here we played naturally recorded purring, rattling, and typical T. oceanicus songs rather than just synthesized pulses. And we did that to flies from both Florida and Hawaii. And we found this really cool pattern that, again, Hawaiian flies were overall more responsive to all three of those different types of T. oceanicus song. So I think if you take those results together, this suggests that the neural auditory tuning and the behavior of the parasitoid fly have evolved in Hawaii in ways that make them more sensitive to a broad array of song types, which likely facilitates them detecting these new host songs as well. And that, of course, could change everything in this system. So in summary then, I hope I've convinced you today that we found at least 4 new morphs of the Pacific field cricket that likely emerged over about the last 10 years, and that each of those male types has a novel sexual signal that is a unique evolutionary solution to the problem of attracting mates, but simultaneously evading this fatal parasite. And over the last 24 cricket generations, we've been really carefully tracking novel morphs, and we've found that they have increased in commonality. They've spread across the Hawaiian Islands. They've even diverged across those islands. And we're seeing that this system is highly dynamic. Everything is in flux. We think now that this rapid evolution is likely facilitated by rather permissive female mating preferences in Hawaii specifically. But we need to keep tracking these things because those natural enemies, the parasitoid fly, might now be responding through evolutionary change in auditory tuning and in behavior. So with that, I will just end by saying thank you so much to all of my co-authors and funding sources, and I am, of course, happy to take any questions that you might have.
Eric está com o microfone desligado.
Sorry, guys. Technical problems. Again, Robin, thank you so much for the amazing talk. Now I'll take the questions from the audience and ask to you. The first one is from Laura Camila. She asked if the silent mutation... Oh, there is the screen for you also.
Yeah, this is a great question. So the silent mutation did spread really, really quickly. In one population, it spread to more than 95% of males in fewer than 20 cricket generations. And this was the very first question, the one that you're asking, was the very first question that we asked ourselves too. How do they actually manage to mate if they don't have their sexual signal? And what we found early on was that there were still a handful of males in each of these populations that were capable of singing. So those silent crickets used satellite behavior. So they would sort of hang out next to a male who was singing the typical song and wait for females to approach that male. And then the silent males would manage to mate with females who had been attracted to the other guy. So that seemed to work for quite some time. I think more recently, things have changed a lot. So silent males are really rare now. But we also had a period of about 6 years where there were no typical singing males in these populations that had gone silent. And so we think that during that time, they must have been using some other mechanism for finding mates. And the most likely one we think is that they actually are sort of hanging out in very small areas. So rather than spreading out across the lawn, they are much closer to each other and they're using searching behaviors. So there are some cool studies that show that these silent males actually move around and search more than a male who's capable of producing a song with. So there might be a combination of satellite behavior and searching behavior that let them be successful.
Okay, thank you. Leonidas did a question, and I don't know if you pop up for you, so I will read. Oh, there's the question. Is there any specific relationship between the vocalization changes in the species and the new environment in which they now live?
Yeah, that's a good question. We are always asking ourselves, because these new types of males seem to be popping up so quickly, like, is there something about the environment that we don't understand? Some pollutant or something that's causing this kind of, seemingly high rate of mutation to happen. We have not found anything other than the fact that in these Hawaiian populations, those are the only places where this parasitoid fly is found. So across the rest of the cricket's range, where they don't produce any new sounds, there's no parasitoid fly. But in Hawaii, there are parasitoid flies across all of these populations, and that's where we're finding these mutations. So we don't know of any other environmental differences. The areas that they're found in are basically just like grassy, disturbed areas. So they're often on university campus lawns, for instance. So there's nothing in particular that differs in terms of the environment that we're aware of that is kind of consistent with these changes in vocalizations.
Related with this question of Leonidas, I took some notes of my own questions.
Yeah.
It's about this dynamic process with the frequency of each trait during the populations.
Yeah.
If they can they respond to any environment or climate or something like this, because they keep fluctuating, right?
Yeah, they do keep fluctuating. I think a lot of that fluctuation, though, is due to new morphs arising that disrupt what the system actually looked like. So, for instance, that handful of years that we showed there, a new morph showed up and started out competing things, and some were dropping and others were doing okay. So there's also a lot of we've done these really cool mesocosm experiments in the field where we built enclosures and loaded them up with crickets of different types. And it seems that the social interactions that the different types of males have with females and with each other really drive some of the success of those different types. So, for instance, if you house silent or purring crickets alone, they don't get parasitized. But if you put typical crickets or other loud new morphs like rattling males in with them, they get parasitized as a byproduct of being close to those males who are singing. So the social interactions between them actually change the selective pressures in ways that are consistent with some of those patterns that we're seeing. We're really interested, though, in those patterns and what all is driving them, right? Because it could be that we're going to start to see, negative frequency dependent selection or something cool like that. But it's too early at this point to know, I think, what's responsible for all of that dynamic change.
Okay, I will pass through the people's questions and then I can keep my own. Okay. Oh, why the crickets from Hawaii changed so much with the fly, and in the other places where the flies are present, these changes are not so prominent.
Yes, great question. So I think that story that I was telling you about female preferences is really, really important to all of this. So the fly is native to North America, and it parasitizes a bunch of different crickets within North America. And none of those crickets have these types of changes in song. that we're aware of, right? No one has reported those kinds of changes. So I think I get this question a lot, like, why don't we see these kinds of changes in North American crickets that have been parasitized by these flies for a really long time and seemingly should be, responding or could respond in similar ways? And I think that the pattern of female preferences that we see in Hawaii versus other places in their range is a really, really key point here, because if those novel songs had arisen, let's say in Australia, females would have discriminated against them and they never would have spread within these populations. So females in other places, just like North American female crickets, would discriminate against a song that didn't have the right properties, right? They don't accept those males. But there's something about this relaxed preference in Hawaii, this permissiveness, that's allowing all of these novel types to stick around and allowing evolution to sort of play with these different types and see which of them is going to be successful over time. So I think it really is something about island populations and the sort of historical, selection that has existed there for less choosy females that's allowing this to happen so quickly.
Great. We have a question from Giovanna. Could be this from Camila. It's okay.
Sure.
Now, are maybe the sensory organs more flexible in anatomy so females can still present variability in mate selection?
Oh, great question. So, in terms of the kind of female sensory system, this is our next big project with Norman Lee. So, we are looking at things like what the structures of the ears actually look like. We're looking at things like the same thing that we did with the flies, essentially, in that last project. So neural and behavioral responses of female crickets across a bunch of different acoustic space. So we're really, really interested in this. We want to know, what do these females hear? Are they actually just discriminating against certain types, or can they not hear certain types? I'll also say that I was telling Erica this before the talk started, that we are doing these very cool plasticity experiments where we rear female crickets and male crickets in different acoustic environments with some of these different novel songs. And if you do that, you end up with, when they become adult, those females that heard the novel song types are more willing to mate with males who produce a novel song type. So we don't know if that is plasticity in sensory structures. We don't know if that's plasticity in preference, right? It could be either of those. But there certainly is something about experience with those songs that leads to changes in their responses to the song. So really, really cool question.
Amazing. I have a question in this related with the previous topic on the environment and thinking about this social environment in the field.
Yeah.
Are these animals territorial, or males present any kind of nest for females to reproduce? Because this could change the distribution of morphs on the environment and modulate this, I don't know, maybe.
Yeah, totally. So they are not traditionally territorial. So they don't actually consistently defend an area, but they will have male-male aggressive interactions with one another. So in the before times, before there were all of these novel types, what you would find in the field was essentially a male every couple of meters, and those males would kind of hunker down in the grass and they'd sing their songs from spaced away from other males. Now you don't find that so much. So they are wandering around more. They will kind of hunker down under the grass, but some of them walk on top of the grass and you can actually encounter them more easily. But male-male aggressive interactions do happen and probably matter a lot for kind of outcomes in these social situations, right? And we don't know if females will do things like base any of their mating decisions on the outcome of male-male aggressive interactions that they've seen or known about in the past. So yeah, there's all kinds of cool social stuff there. I just did a really fun experiment with my animal behavior class where the students set up male-male aggressive interactions between typical males and then between males who have these novel song types. And one of the cool things that we found was that the males that have the quiet novel song types actually get into more intensive fights with each other because they
It
don't have
would
that
be my tip. It would be my tip.
to like bring to kind of tone things down, right? They can't just say like, I'm big, go away. So yeah, they end up getting into like more intensive fights. And so that kind of thing could play a big role in who's successful in nature and who's not.
Amazing. Okay. More questions? Okay. What about sensory modalities? For example,
Yes.
either carbons in exoskeleton of the crickets.
Yeah, awesome question. So they do have contact pheromones, so cuticular hydrocarbons that they can use to tell the difference between the sexes. Traditionally, those are thought of as only contact, so they have to antenate to pick that up. But I've actually talked to some chemical ecologists who think that they're probably also laying those down on the grass, right? So I think this is one way that they are able to figure out where the local high density areas of crickets are. So if you stick around in the grass that smells appropriate, you're more likely to run into other individuals than if you venture out into grass where nobody's been hanging out. And we know from Nathan Bailey's lab that there are some differences in the cuticular hydrocarbons of typical crickets and silent crickets. Silent males actually have feminized cuticular hydrocarbons, so they smell more like a female than a typical male does, which might actually be one of the reasons that you get those more intensive fights. So you'll actually see females fight with silent males during courtship, like the male is courting her and she'll kick him away. And so we think that kind of increased rate of aggression toward these novel male types might be because of some changes in cuticular hydrocarbons. To our knowledge, though, that hydrocarbon, like those contact pheromones, are not able to sort of override the song in terms of preference. So the song is really still how they are making these mating decisions. And we haven't looked at cuticular hydrocarbons in the novel male types yet. So it's entirely possible that there's some other, combination of things that females are getting during those courtship interactions.
Amazing. Some more questions from the chat, guys. Let me see. Second. Okay, maybe no. Let me see my own. Oh, thank you for the talk. Such a great opportunity to hear about this subject. And I'm sad. And I have a lot of questions, but I can send to. Oh, Laura Camila. Another question. There is a chance that the typical song disappear given that females prefer novel songs.
Yeah, great question. So we do have some populations where there are no typical males anymore. For instance, there's a population on the island of Oahu where only purring males are found. If you were to give a female the option of mating with a typical male versus these other types, she'll always take the typical male first, right? So there's still a preference for typical over the other types. It's just that these females in Hawaii are more willing to accept the novel types than the females from elsewhere. So I think unless the parasitoid fly only attacks those typical males and drives them out, female preference would still maintain some of those typical males in the population. In fact, in those mesocosm studies that we did, we looked at fitness of typical versus purring males when they were in competition with each other. And although most females mated with all of the males that were available to them, they actually had more offspring with the typical males than they did with the purring males. So there might be some cool, cryptic female choice happening here too. So I think those typical males are still going to have more offspring and are still going to be preferred by females if they're given a choice. But you're going to see this sort of creep up of novel types because they're more protected from the parasitoid, at least for now. We'll see what happens with the fly.
Amazing. That would be my next question about the offspring when you use pairs from different morphs, phenotypes. What's how is the phenotype of the offspring? So
Yeah. Yeah.
answer.
So when we bring these guys into the lab, when we find a new morph, the first thing that we do is do some breeding experiments and try to figure out if they'll breed pure, right? So we do have pure breeding lines of these different types. Some of them are more complicated to get to pure breed. So for instance, rattling disappears pretty quickly in our lab populations. And I'll give away some new data. We have some very, very new genomic data that suggests that purring is likely something that is located on the X chromosome, similar to silence, but rattling actually seems to be autosomal and to have kind of multiple different like peaks on different chromosomes. So it's a polygenic trait, we think, which might explain why it's more difficult to pure breed in the lab. So we have some genomic data that will be coming out soon. We also have like field data from the last 6 years essentially gives us information about what happens when these morphs mate with one another, right? So when we end up with like a mom who, had mated with a flatwing male, but then ends up with daughters who do X, Y, and Z. Essentially, what we're seeing in the field now is that we don't have just pure individual types of males anymore. We've got crosses, we've got back crosses, we have intermediates between different morph types. So that figure of morphs interacting over time is getting way more complicated because they're all interbreeding and we're ending up with hybrids of all kinds of different types. So they will there's a genetic basis to each of these things, but what we're seeing in nature now is that they're all interbreeding with everyone, and so we're getting all kinds of intermediate forms between those morphs over time. So it's getting more complicated, but more fun.
Okay, thank you so much. So I think that's it. We passed the 15 minutes. It was an amazing talk, and thank you again for accept our invitation
Of course.
to be here with us.
Yeah, thank you so much for having me. It was really fun.
Okay, and guys, next Thursday, we have another question. Terça-feira a gente tem outra palestra. E for people that do not sign the form, if you want a certificate of this presentation, you can sign the form here. And I think that's it. This presentation will be available in the YouTube channel from Instituto de Biociências. And that's it. Thank you for being here with us. Bye.
Thanks.